this Tambun, but also other place in Nan. It was investigating the antimicrobial resistance, and with the project Farm Resist that involves Switzerland, Thailand, and France. And we have also to thank the support of the involvement of Faculty of Veterinary Technology with Dr. Anamika, and the Faculty of Medicine, Sir-Village Hospital, Faculty of Tropical Medicine, and the Marseille Institute of the Health Institute of Medicine, and on the Faculty of Bern on Lausanne also. We did a lot of investigation questionnaires, but we investigated the exposure to antibiotic, especially the antimicrobial resistant genes in the fecal samples collected for farmers and their family, and from pigs, chickens, and also wildlife, the rodents, but along a gradient of ecological gradient, but also a gradient of intensive farming, small and medium-sized farming of chickens and pigs. We did a lot, and thanks for this project, we fully sequenced 1,000 strains of bacterial, and we did a lot of genetic analysis, phenotypic analysis, which can be help also to better understand the use and the impact of antimicrobial resistant genes, especially the misuse in medium and small farms, so to improve actually the use of antibiotics. A third one would be about the chigger mites and scrub typhus, and thanks to the involvement of the public health, the Ministry of Public Health, and Dr. Hawadi that we speak also to today, and also with the Faculty of Tropical Medicine. And here we were involved in terms of developing a better innovative tool for chigger mites identification, but also follow-up scrub typhus in the Sentong. Similarly, a new project that starting in 2020, and it was very fascinating project. But this afternoon, Dr. Michel and Dr. Hanan will discuss, and our colleague will discuss a little bit more about this important project using dogs as sentinels in terms of exposure to potentially zoonotic disease, and developing innovative tools with collars and follow up the infection, but also with questionnaire. But we will know better this afternoon. Innovation also and follow up in terms of One Health, which is the mosquito and bird-borne disease, with a project supporting by Montpellier University and with IRD, and involving Rodolphe from this IRD, but also the Faculty of Tropical Medicine, and collecting mosquitoes and developing sequencing to this bird-borne disease. But we will know today also more about this project. Exposome with pesticides, and we thanks also for this project that was actually coming up because of the community. The community asked to develop this project, and we follow up their exposure to glyphosate, but also develop some approach in terms of participatory mapping of the use of pesticides in relation with the cropping, and after also developing some knowledge, attitude, practice with thanks to the help with Dr. Flavie, but also Dr. Pongshai from Chulalongkorn University, and Dr. Tanaradi for the Faculty of Science, Social Sciences of Kasetsart University. So you see, all the time try to put people together. And the new project is a virus spillover at the interface between bats, rodents, domestic animals, dogs, and humans, starting in Sentong. This project was supported by the new project INER, but also by our project. So thanks to Dr. Mariadek, he will be there. This project is in Thailand, but also in Laos, and thanks to Eric Deharo, but also Eric Leroy. And we have also a big investment of Faculty of VetTech and Faculty of Tropical Medicine, MIDOL. And this will be part of a new master thesis of song that will be there, and starting work a lot on this, on questionnaire focus group, bat song recorders, camera trapping, collecting back guano. But this we will present more next year about the outcome of this project. So really try to really develop a One Health project with social ecology, better to understand the sharing pathogen between humans, dogs, rodents, bats, but also other wildlife, and within sharing habitats. So better understand the risk. So what is the danger related to the behavior and related to the vulnerability of the system? So finally, it's like this, try to work on this. So with participatory, with community involvement, on collecting a lot of data, and after more disseminating. So we are on this process to work like this. So mostly to have a better formulation, protocol design, implementation, stakeholder engagement, ethical engagement, realization, communication, recommendation, and working all together, communities, departments, ministries, and academics, and to better disseminate, advocate, and of course to have a better documentation at the One Health perspective. This is not only this kind of pilot study site of observatory. Dr. Kitty Ponk will present also a new observatory on biodiversity and disease ecology, and I hope that some of the people from Faculty of Veterinary Technology will take. So I will finish because it will be a long introduction, but we can go there to this project objective, and with several talk today that are really fitting with this objective of the innovative animal health TACA. First, in innovative research, we will have a Dr. Paisin. Even if he's not really involved in this, we think that is really important in terms to better understand the ecological interface. We have the Dr. Alexis and Professor Wilson Wen from Taiwan and respectively also from Barcelona to really work more on the helminthology, the importance of helminth and helminth-borne disease for wildlife and for humans. But on rodent-borne disease, we will have Dr. Pipat from Princeton Clay University, Dr. Maria Gzek from France, and Dr. Alice from Belgium, and also a new project on all pellets by Dr. Julian. So using actually like a nature-based solution for better understand biodiversity and biodiversity change, and with some impact on disease ecology. Observatory, I already told that, with a disease ecology observatory by Dr. Kitty Ponk. Innovative tool, we will have vector-borne disease this morning with Dr. Rawadi and Dr. Rodolphe, and dog health at the interface this afternoon with Dr. Michel, Professor Anand, Dr. Putu Heidi, and Johan, and traditional veterinary knowledge with Dr. Nicola. And I hope that we will start the conference with one important project on One Health and conservation by Professor Wayan, and we will finish the day with another focus on the translation with Dr. Claire on One Health, law and governance. Thank you, and we can start.
Thank you, Professor Dr. Serge Moran, for the presentation. And now I would like to if anyone has any questions for Dr. Moran.
Yeah.
Yes, sir. Or if you have any questions, you can leave the message in the chat box.
Yes. Sorry, I think that Professor Wayan is not there. So is Professor Wayan is there? He's not there. So if we have some questions, please be free to ask some question. If not, we will go to our next speakers, and we hope that Dr. Ryan will be there.
Dr. Paisin Lee Lecturer. Yes. So if there's no questions, is there any questions on the chat box?
I didn't see any questions, but I see some people saying good morning to everybody.
Yes.
Maybe Seven, you want to ask a question?
Okay. Good morning, everyone. Can I ask a question? You hear me?
Yes, for sure, Dr. Seven. Very happy to have you there.
Good morning, everyone. It's nice to hear everyone again. And Serge, that's a great talk. So my question is regarding this, we call it One Health goal, and for people stakeholder who involved in the stakeholder involved in these projects, is there any decisive or any importance message you learn from the local peoples and the local stakeholders?
It's an important question, and you're right. That's why we will have a presentation this afternoon by Dr. Nicola about the traditional veterinary knowledge by the local people, a just starting project that will involve also Professor Piboun at the Faculty of Veterinary Technology. We hope that it will start well next year. Actually, Nicola was there and just leave a couple of days ago, cannot be directly there. This is one thing. So on the traditional veterinary knowledge. The other one, we work a lot on thanks to Song, but thanks also to Dr. Tanaradi and all of the students of social sciences. And there was also some student involved from the Faculty of Veterinary Technology, is also to better investigate, understand the traditional ecological knowledge. So, and who has how it's processed and how we can also record all of this. And one important thing that they show, and it is not really unusual, there is a loss also of this traditional knowledge. So I would say that all the young generation now with the school, with the mobile phone, and they have more activities on attracted by the cities, and they are not going so much on the field. They are not going so much to be trained by their parents, either the mother or the father, or either the grandpa and the grandma, and they are losing very fast the knowledge. I can say, for example, you have the people, I would say 40 years and older, they can know a lot on plants, on trees, on animals. If you ask for a child of 10 years old, when you see this, he cannot name the plant, the thing. He doesn't know how to use them. And I think that this is something important, and they realize this, and especially at the Tambud, because one of the things that is important for the sub-district level is also to maintain the culture. And the culture is also the culture of the nature of what they know. So we need better to be involved with them on this.
Thank you very much, Serge.
Thank you, Dr. Ho Wing Chen, for the questions. It's very nice, pleasure to have you here. And so, if there are any more questions, and on the message box, I think
No.
no.
Okay. I think we can go. We will have more time to discuss for the presentation, and if Professor Wayan thinks we can manage. I see that Dr. Paisin is there, so maybe he can share his presentation and start his talk. Welcome to Dr. Paisin, that is doing his PhD thesis at Chulalongkorn University. And I will leave the floor for him. Dr. Paisin.
Okay, thank you, Professor Dr. Serge Moran, for your presentation. So I would like to invite the next presentation, Dr. Paisin, lecturer. He is from Faculty of Veterinary Medicine, Chulalongkorn University, Thailand, and he's going to present in the topic of wildlife interface at waste disposal, which is a quick look to wildlife, domestic animal or human interface at solid waste disposal site, emerging infection disease, and we will have him soon. So before that, the program will be next with Assistant Professor Dr. Kittipong Chai Siri. And later will be Dr. Ravadi Kamler. And then finish in the morning part with Dr. Rodolphe Haumet.
Nice. So we have now
So I think we.
Dr. Paisin that's
Yes.
starting to share.
Yep.
Okay, we have some problem.
We try to open his slide video.
Okay. So just a little minute. So to a little technical problem, just to share the slide.
So before the Dr. Paisin presentation, I would like to invite you all for the next, for in the afternoon presentation will be Dr. Michelle Garen Wichit-Tischstein from Seyrat, France, in the topic of social ecology of dog-borne zoonotic disease in SE Asia. And then in the afternoon, next presentation will be from Professor Anand Poonpom from the Faculty of Computer Engineering, Kasetsat University, in the topic of tracking movements of dogs in rural landscape of SEA Asia. After that will be Dr. Heri Wijayanto from University Gadjah Mada, Indonesia, in the topic of the relationship between the dog's home rank and zoonotic disease transmission in Karangsem, Bali.
All participants, thank you for having me in the third Veterinary Technology and Nursing International Seminar. My name is Paisin, lecturer from Chulalongkorn University. Today I'm going to present my preliminary results from my research in the topic of a quick look to wildlife domestic animal-human interface at solid waste site. In the past few decades, after global eradication of smallpox and triviality of infectious diseases, we are in the great time of encountering the global health threat of emerging infectious diseases. 3/4 of those are zoonotic diseases, and 60% of infectious diseases in humans. The recent and the new ones are originated in animals. These zoonoses have caused over 2.5 billion cases and 2.7 million discoveries each year. The EID also caused significant loss of economy and social problems, threatened global food security and biodiversity as a consequence. We put huge efforts on studying the origin of EIDs, as 60% of EID originate in animals, 71.8% were found to have wildlife genetic origin, suggesting that 78.2% to jump to human from domestic animals. However, 13 zoonotic diseases were linked to 2.4 out of 2.5 billion human cases, either directly or indirectly to food consumption and vector-borne, of which 9 diseases implicated livestock illness. It is suggesting that a great number of wildlife zoonotic pathogens find their way to humans through domestic circumstances. The term of wildlife here would consider domestic, captive managed, and farm wildlife, and free roaming, and those can form an area of interface altogether with domestic animals. While human composes 36% of global mammalian biomass, and another more than 60% are livestock. Less than 4% of this, more than 50% are marine species, are wildlife. A large portion of wildlife that may contribute to be an intermediary of spillover is polydomestic or synaptic species. Polydomestic wildlife are defined as animals adapting to human landscapes and living in close proximity to people, including animals brought into human habitation as food. Nonetheless, those synanthropic animals are typically from the rodent, primate, carnivore, herbivore, bird, and taxons, and are the most important from the perspective of zoonosis and EID's view. The study why these species are attracted to human landscape and what is the composition among these species is a key to understand its relationship to human, including disease we share. As human population growth and urban development, there are more human-dominated landscapes. Every day, a person generates an average of 0.74 kilograms of solid waste as estimated by the World Bank. We produce a total of 2 billion tons annually and may increase to 3.4 by 2050. More than 30% of municipal solid waste were not managed in an environmental friendly manner and remain at the site. This creates an unsanitary and unfavorable condition to the public. As a result, we needed more land to dispose them, and in general, we often built up places far from public and into the forest. Nearly half of total amount of organic waste This may provide additional nutritive source that attract wandering animals, either domestic animals and wildlife. Some types of waste provide shelter, burial, and breeding sites for several pathogen vectors and reservoirs. This can alter their ecological properties and behavior. Furthermore, kitchen waste and animal waste contribute many pathogens to the environment. All these factors are considerable to create area of domestic animal and wildlife interface and change rate of pathogen transmission among them. Although it is mentioned about health problems related to solid waste management, most of them focus on the consequence of gas emission, leachate, and flood contamination. Study about infectious diseases is less obvious, and therefore, information about the structure of animal community and the relationship to risk of disease emergence is still lacking. Number of species have been reported at solid waste sites, although they did not demonstrate with regard of infectious diseases. Some studies suggested a relationship between dog rabies and municipal solid waste management. Asymptomatic behavior of dogs and wild carnivores may contribute to a maintenance of this fatal disease for humans. The presence of non-human primates can involve in many genetically related pathogens such as herpesvirus. Although there were rare reports, pathogens in humans can also invade to primate populations such as tuberculosis and influenza. Solid waste sites containing kitchen waste and civil may provide hazardous animal pathogens such as African swine fever virus that is considerably fatal to wild population. Although it is not zoonotic, such disease may disrupt the process in ecosystem and induce catastrophic consequences to humans. As the most diverse taxa of animals in the world, rodents are associated with more than 60 zoonotic diseases. The number shown in this slide provides information of the magnitude of important zoonotic pathogens in the rodents and other small mammals in East and Southeast Asia. Because of some biological properties of these small mammals, such as high reproductive rate and relatively short lifespan, they have been used as a model for a study relationship between environmental change, climate change, variation in community properties, and the prevalence of rodent-borne diseases. Several rodent-borne pathogens have high host plasticity, meaning that they have wide host range. Rodent abundance fluctuates during the years and between seasons upon food availability, so this may affect the prevalence of disease. The study found that the infection rate of some zoonotic pathogens were sensitive to the diversity and composition. However, such study in the solid waste site circumstance still need more investigation. So, this study arrived to investigate a presence of wildlife and domestic animals at solid waste site near the forest. The study was conducted at solid waste site near Tongpayin Khao Yai Forest Complex. The survey included a camera trap survey for medium to large mammals and a capture mark recapture protocol for small mammals. In medium to large mammal survey, we used a camera trap survey. This survey was conducted at the solid waste site only. The number of camera trap stations at each site were up on the site of solid waste, and approximately 6 to 15 camera traps were prepared. The duration of trapping was at least 30 days but can be adjusted to reach a target of more than 100 trap days. We expect the camera trap to capture medium to large size mammal as shown in this slide. The survey of small mammal was different from camera trap survey. This survey was done at the solid waste site and also for other land use types around but within 2 kilometers of corresponding solid waste site, including human modified habitats such as field of paddy field or oaks, and natural habitats such as forest. Wild-made live trap view were arranged in a grid. At the solid waste site, traps were placed throughout any accessible area where animals may present. For other land use, the grid consists of 36 tracts of 6 by 6 area were used, and each tract was 20 meters apart. We found 177 sub-districts belonging to 5 provinces were connecting to Dom Phayan Khao Yai Forest Complex or its forest vicinity. 157 local administrative offices were set to govern around this area. However, there were only 48 solid waste sites under supervision of these LAO. Three sites, BRRO1, NRSO1, and NRSO8, were selected for studying animal ecology. The information shown in the table included number of LAO using each solid waste site, population number of local residents, size of solid waste site perimeter, average daily amounts of waste, and waste disposal techniques. BRRO1 was responsible for about 10,000 people that created 7 tons of garbage yearly. The disposal method was just open dumping. The site located near a large reservoir and Papaya National Park. However, most of the land were agricultural area. The NRSO1 located in a large isolated community forest surrounded by agricultural land. There were 6,000 people creating 4.3 tons of garbage yearly. The LAO used open dumping as a disposal method. The site was located near the Papaya National Park. The NRSO8 was by 2 local administrative offices and was responsible for about 10,000 residents. This area was famous for tourism, so a number of resorts and hotels and tourists be far and added up the amount of solid waste in this area. The disposal techniques were open dumping plus incineration, and this solid waste site located near the Khao Yai National Park. The position and number of camera traps were shown in this picture.
No, take after my
As to detect the animal who was
plug.
in the solid waste site, the camera traps were set at the border of solid waste site.
After
The duration of working period
the
was
bees,
more than a month for each site.
students.
I would say
Domestic and wild mammals were
Question
tested at each study site.
to go on.
Large protected and endangered species according to IUCN Red List was
The
found
next
at ISO 8.
little 5
Other
minutes.
wildlife included carnivores such as small Indian civet, small Asian mongoose, Asian fox civet, and green shepherd. Wild boar was also found at BRRO1. Small mammals such as Bandicoota and reptile species can be found from it, grouse, snail, and tree chew. Domestic animals included dogs, cats, and cattle. Many rodent species were trapped at solid waste sites dominated by Rhatus complex and Rhatus excellens. Most species can also be found, as well as Bandicoota species, but in a small portion. In the agricultural area, the most prevalent species were musk cervicolor, followed by Rhatus complex. In the forest and rangeland, Rhatus complex can be found in addition to Maxomys sorifers. Non-marine species Amenites batmorii and non-rodent Papaya blancheri was commonly occurred in the forest vicinity. Since the occurrence of both domestic and wild animals have been found at solid waste sites, it was possible that the area of interface has been observed. This may increase probability of pathogen spillover across species. For example, a presence of wild boar may lead to African swine fever virus spread from food waste contaminated in solid waste site into wild population. Since the pathogen was very strong against harsh environmental conditions and may remain in solid waste site for a long time, a symbiotic occurrence between wild and domestic carnivore may persuade the circulation and spread of rabies at solid waste site. The disease may complicate the conservation of wildlife, especially for small populations such as large spotted civet. Disease may spread by this synanthropic wildlife into sedentary wildlife population in the wild. However, more study is needed to conduct to understand the relationship between host species ecology and pathogen community at the solid waste site. Solid waste site was also support the existence of the most diverse mammal species, the rodent. The species diversity of rodent was relatively higher compared to those in modified agricultural land and cropland. The species diversity may be high in the nearby forest. However, the community composition and species abundance were different. It was interesting to further investigate the dynamic of small mammal community on disease transmission across these landscapes. In conclusion, this was only a preliminary result and further analysis is having to be done. However, it seems that solid waste site may provide several basic needs for wildlife and domestic animals such as food, shelter, and breeding sites. Future research on disease transmission, prevention, and control should be done in the integrative way with improving waste management policy. I would like to thank our organization and colleagues who have supported me in this study, and thank you very much for all participants for your attention.
Presentation. So it's very able topic. And is there any questions from audience? Is there any questions from audience, or you can leave the message in the chat box, or if you can raise your hand for the questions.
So please don't be afraid. You can speak, and I think I would like first to thank Paisin for this very amazing and very interesting talk. I'm sure that there will be some questions. Okay, so I will have one question for you, Dr. Paisin. My question is how to, because your work is really at the interface between the conservation with the national park, but also with all of these diseases and the wildlife, but also domestic animals, and with the communities and with the problem of the waste management. How you imagine, or you want to translate the result of your important studies to the communities, but maybe also to a better management at the level of the Tambun of the district of the West.
Well, thank you very much, Texas, for your question. So this is one of my interesting perspective that I would like to draw up some conclusion and recommendation that I'm going to transfer to local people. That is not only for the just the layman or just a local resident, but also for the local government as well. And that is not for only the health perspective, but also the conservation perspective, because you know that the health test may not impact only for people, but also for animals as well, especially for the wildlife. So there's a lot of things to be done to like draw up these conclusions. And I think the very preliminary result might be very useful for talk to the local government. So they might have some like campaign to talk to their local people about the waste management. For example, they might ask people like to try to reduce the amount the waste that they generate. So they might it might link to the waste generation, and if they have improved their waste management, and I'm pretty sure that the risk of the waste at the attracting site for the animals might be reduced as well. This is my idea, my very first idea about that.
Thank you very much. Thank you. I see that Professor Pizzanou have some two questions. Maybe I invite him to speak and to ask directly the question to Dr. Paisin.
There's a question for Dr. Paisin from in the chat box. The first is from Dr. Pisanu. He asks, what are the criteria to select the solid waste land in this study?
Thank you for the question, Dr. Pisno. So actually, this is just only the 3 sites that I have been accessed, but and I have more sites to be investigated in the near term. In the meantime, the criteria actually in one of my study fairs, I conduct the multi-criteria decision analysis to select the appropriate, the suitable site for studying. I define the suitable site as the area that can promote the interface. So the criteria might come from the expert opinion. I try to like capture the idea from the expert opinion first to do the multi-criteria decision analysis, and then I'll use the information from all solid waste sites around the Dong Phien Khao Yai forest complex to input and calculate and ranking them. But I haven't shown in my talk today, and I can show you maybe next time if I have opportunities.
Sure.
But I can say that most of them include the something like the distance from the forest, the occurrence data of the livestock or domestic animal population, the human population, because it had exactly linked to the waste that they generated. So this is some of the criteria that I use.
Thank you.
Then there is one more question from Dr. Pisanu. What are the important messages that you would like to share to the people who stay at the interface area?
For this question, the thing that the people should know is that we generate the waste, and the waste need to be managed, and the way that we manage the waste might have a huge impact on ecology, and it might create the risk of pathogen spillover among us people and also other animals, and it might create a possibility to create the new pathogen that can return to us later. So please mind about the waste management or the waste that we generate. This is the main message that I would like to say to people.
Thank you, Dr. Pasin. And there is one last question from Dr. Julian. Thanks for the presentation, and she really enjoyed it. And there are the questions is, were the rodent molecular identified If so, why mice have been identified at the species level but not rodent, raccoons? Sorry.
Yes, right now, I use the reference, the morphological criteria to identify first, but there are some species that I have to do the molecular identification. So it's not just in this phase, but yeah, I would like I may do the test, the molecular test identification and for both the moose and lettuce species, and the data, the information might be more precise.
And I think I have one more question from the message box for you. The question is, how can you identify the proportion of feral dogs versus own dogs?
So because we have stay for an extending period at East Solid Waste Science, and we can like see the how the dog live in that place. So it cannot be sure for all the dogs, but there both field observation and also the camera tab might say something about how to differentiate of the own dog and the feral dog. But actually, for further analysis, the role of these two group of dogs might not be much different, because they stay like for a long time, and they might act as have the same role for the disease transmission and maintenance. I think so. Sometimes we will not have to like try to identify the own free roaming dogs to the feral dogs.
Yes, and we have one attendant who raised a hand to ask questions. Please, Dr. Michael.
Yeah, thank you. Actually, you just read my question, so I think Paisin has already answered to it. I think the talk is excellent. It's raising a lot of questions about the interface. My question was specifically to say that for the dogs that are actually visiting these areas, it makes a difference in terms of breed species, whether they are true feral dogs or whether they are owned dogs that are free roaming, because then the contact patterns with potential humans, with their host, would be different. So discriminating exactly which dog are actually free roaming but are owned coming from households, from the dogs that do have very little contact with the human populations in the area, it makes a difference in terms of maintenance and epidemiological significance. Having said that, I know it's very difficult, and that's actually one of the things we're trying to do using camera traps for identifying specific dogs. So we'll give some insight about this in the afternoon.
Dr.
Yeah, thank you very much, Dr. Michelle. This is so cool that you so yeah, one of the different thing that the own dog, they might come back to the community and maybe like transmit something that they have contact with the domestic animal, that of domestic pet at the household and the people as well. So that is the main difference. And yeah, the work that your team have done in Nan, so putting the collar to the dogs, it might have, I might think about how to like apply these techniques to
identify both the own dog, own free roaming dog, and also the true feral dog. So thank you very much.
Most welcome.
Thank you very much, Michel and Paisin. If there's some other questions. Actually, we don't have Professor Wayan on the line, so if not, we will continue. But first, I would like to really thank Paisin for his talk and especially for his important study. I think it's the first time, and I read a lot, and it's the first time that one person starting this kind of important work to look at what ecologists try to avoid, the garbage site. And because most of the ecologists want to work on the national park, on the nature, and you decided to work there. And I was very happy also to join one of your field work. And I can say that I was also very amazed by the protocol you designed, and very well built with development also of a nice way to take data and to get the data. And the quality of this, which is also involve a huge investment in terms of work on the field and after work in the lab. And your study will be one of the important study that will be publicized very soon. I hope in the next year you can have some starting papers, because in the world, I would think you will be one of the leaders to discuss about and to highlight and to emphasize of this important, Michel, hotspot interface, and especially for disease contact and transmission, but also very important in terms of conservation biology, because not only for the human health, but it's also for the wildlife health. Thank you very much, Dr. Paisin. Thank you, and good luck to continue your important work.
Thank you, Dr. Paisin and lecturer, for your presentation, and it's very interesting. Professor Moran said that it's very interesting that you do with the waste disposable. Next, we would like to invite Assistant Professor Dr. Kittipong Chaisiri for presentation in the topic of projecting a long-term observation on biodiversity and disease ecology at Biodiversity Kanchanaburi campus. And Professor Kittipong from Faculty of Tropical Medicine, Mahidol University, Thailand, if ready, and I would take the stage for him.
So first of all, I would like to thank the VetTech International Seminar organizers for your kind invitations that allowing me to present our plan and proposal on the collaborative project on the long-term biodiversity observations. And this is ecology study that we are focusing the three main animal groups, which are rodents, bats, and birds, and also their parasites and pathogens that may carry with these three groups of animal at Mahidol University Kanchanaburi campus, or we call MUCA. So what I should start with first, I would like to introduce a bit about like a coordinated situation of current global environmental problem with like unavoidably linked to health and well-being of the earth, wildlife, animal, and human. So nowadays, scientists and researchers agree that we are moving into a new era that called Anthropocene, which human footprints have covered everywhere on the planet. And here, our activities alter many conditions and processes to the Mother Earth. These are resulting in like a big changes of several events, and some events even unable or nearly impossible to return, such as like fast environmental changes, more and more erosions and sedimentations occur on the surface of the earth, including also some other like climate variability, occurrence of like new substance that dominating the world at the moment, like plastic, concrete, and also air particulate matter. And also the important global biodiversity loss issue as well, that showing the big effect at the moment. So these changes are also faster than we thought, and this could be the fastest phenomenon in the history of mankind, probably. For example, in the last 60 years, since like 1950th century, human populations and also our activities sharply increased during this just short period of time, including like, for example, explosion of human population number, particularly those humans living in the urban areas, increasing of like primary energy consumptions, water consumptions, construction of like large dams, many buildings as well on the planet, paper productions a lot, and also heavy expansion of like transportation system, improvement of telecommunications, and also tourism across the world as well. These consequently and largely affect global environment and also ecosystems in like many ways, such as like rising of carbon dioxide, nitrous oxide, and methane emissions to the atmosphere. Also, other events are prominent, such as like increasing of surface temperature, ocean acidifications, forest loss, and also habitat changes to domesticated land, urbanized area, and also big cities around the world. So these things lead to like a terrestrial and also marine biosphere degradation, and also big issue of the global biodiversity loss. The result of the latest Living Planet report from WWF and also CSL shows different rates of biodiversity loss through the absolute declines of living planet indices in every continent on Earth, with an average of like 68% reduction rate in the global aspect. One of like several effects from biodiversity degradation is that the loss of regulatory functions to balance host and parasite interface at many levels, like local, regional, and also the global scales. It has been proven by many scientists and also researchers that biodiversity loss causes the loss of an important ecosystem service to buffer the spreading of infectious disease among the plants, wildlife, animals, and also human as well. From the upper figure here, this shows geographical hotspot of like IUCN Red List threatened species as like already extinct species or probably nearly extinct in the future, while the other figure at the bottom represent like similar patterns, but this time in the form of like geographical hotspot of zoonotic emerging infectious disease, which obviously showing the same regions where majority of the threat of biodiversity occurred. So there are some link between the biodiversity loss and also increasing of like emerging infectious disease here. So as a researchers in the field of health and life science, ecologist, naturalist, and also expert in other related fields as well, monitoring of like biodiversity and also understanding on disease ecology are really important to tackle the problem of emerging and re-emerging disease. However, there are some like challenges and obstacles that still prevent us to achieve the goal, such as like how to link academic and research trainings among local, national, and also international institutions as well. Also, how to encourage people sharing their data open data to the public and also promoting citizen science approach for sustainably monitoring in the long-term event. Some initial and simple steps that could be proposed to start the process, such as like projects that collaborate on the long-term observations rather than to do a cross-sectional study, and also create like training of the next generations, younger generation, and also workshops to share experience, protocols, and also techniques among the groups that working in the same field as well. From our own experience since like 2008 onward until now, the 3 projects coordinated by Professor Serge Morant have been carried out in the field of like biodiversity, infectious diseases, and also One Health in Southeast Asia by using like mainly rodent biology and also rodent-borne disease as the model. Although the research project series meet successions and be able to deliver fruitful scientific outcomes in terms of like number of publications and also contribution to the local and international academics, but we still haven't succeeded much in the angle of biodiversity monitoring and also understanding the real disease ecology subject, which require longitudinal study, extensive data collections, and also collaboration from different expertise in this field. So we would like to initially create a platform for sustainable project on biodiversity and disease ecology in Thailand through like combination and working in parallel between the long-term annual census activity to survey biodiversity and population dynamic of host and the parasite, as well as the research and academic activities through the trainings and also workshops. So we believe that the activities between the 2 platforms can fulfill and support each other in the long run as well. So here in Mahidol University, Kanchanaburi campus on Mukha, we have a nice resources and facilities in the area of almost 10 kilometers square. Most of the territory in the campus is covered by forest and limestone mountain landscapes. These providing like great resources of wildlife and biodiversity to explore. Moreover, several academic institutions join and run their activities here as well. For example, you have the Tropical Medicine Research Center, the Faculty of like Veterinary Science, Wildlife and Livestock Hospital, and also you have the Conservation Biology Bachelor Degree program as well running there. So these make like the Mukha quite a great place to start a mini research or academic cluster on the biodiversity and the disease ecology right now. So we plan to start with the study on the 3 animal host group, which are like rodents, bats, and birds, or we call RBB. At the first stage, we would like to construct a baseline data of these animal groups and their parasite and pathogens that the animal may carry with them. Also, in the long run, we can monitor like their population dynamics and also interactions in the context of like disease ecology through a long-term study approach. Yeah. So we plan that every year we will hold an annual census activity by recruitment of like experts in the field of rodents, bats, and birds, and other wildlife taxonomy and ecology as well, to monitor the animal diversity, population status using like classical observation techniques, trapping with like mainly use capture, mark, and recapture approach, and also using camera trap as well, for instance. Also, biological specimens are able to collect it as well, such as like oral swabs, rectal swab, and also ectoparasite investigations on the body. And all of this will be preserved in the biobanking platform for using further in the parasite and pathogen laboratory investigations as well. On the other angle, associated environmental data, such as like habitat types, land use information, geographical information or GIS, and also meteorological data will be acquired from like available sources on internet or geographical informatics online platform as well. So these will be brought together in the central database in terms of like data warehouse, and also this will be kept for further data analysis and implementations. For example, in the field, how we can monitor the animal populations. So in practice, we can partition the study area in MUCA into like different zones or grids, if you like, and conduct animal survey appropriately regarding to each animal capture techniques, and also trapping effort for each of the 3 animal groups as well. At the current stage, we have like identified stakeholders and key collaborators to start this off this year and also into the next year as well. This consisting of like academic institutions, universities, organizations in ministry, and also international cooperative agents as well, as described here in the list. Another important step, we plan to organize an annual academic activity through like a field-based course or workshop or summer camp or winter camp, if you like, for example, to roll on the disease ecology study subject, probably next year in 2022, for example. So our target participants will be postgraduate students who interested in like disease ecology, and probably open to like public for others who interested in this field as well, to join in like intensive activity from field to lab, and also data analysis in a range of like 10 days to 2 weeks period, for example. Here is the example of the 5 main modules and activities that we could plan to fit the course overview. These including sessions on the ethical considerations for conducting research involving animals and pathogens. Also, we could guide the participants on study planning and also how to prepare for research protocol in the disease ecology subject. On the session of wildlife host observations, the participant will learn and practice the methods on animal trapping, animal observation techniques, animal handling, and also specimen collection from the experts in each animal groups as well. In terms of parasite and pathogens and also vector, participants will have a chance to learn and practice laboratory techniques for parasite and pathogen investigation in the lab. On the environmental side, we will have like a session for guide the participants how to acquire environmental data, such as like using GIS application in the disease ecology as well for data analysis. And in the final sessions about the database management, data quality control, and also data analysis practicing in disease ecology subject, such as like you can do many things like diversity analysis, host parasite network analysis, statistical modeling, GIS mapping, and the spatial analysis, for instance. And here we are at the final slide. This shows the expected outcome of the project. So we could have like a research network on biodiversity and disease ecology in the form of mini research cluster, mini academic cluster, for example. So this will give an opportunity to propose for a big grant, either for like application to the domestic grant provider or international grant opportunity in the future as well. Also, we hope that we can have like every year in commitment to conduct annual census activities and also workshops on the biodiversity and disease ecology as well. So this will give opportunity to get like academic recognition as well. Also, the long-term monitoring data will give us a big data set of biodiversity for further use in the research. This certainly gives opportunity to create novel knowledge and also increase the publications in the scientific journals as well. So that's it. Thank you very much for your attention.
Montpellier from Europe. And what also important that we, as you mentioned, is disease ecology, but is really grounded on conservation biology. And thanks with the group of conservation biology there, which means that you attract also and you focus mostly on the knowledge of biodiversity, ecology, dynamics regarding reservoirs, regarding vectors. So you fulfill a gap between what we name the surveillance system that are only regarding at collecting wildlife, but really to understand this dynamic on the long term. So I would like to thank you for this. Any question for the start?
If you have questions, you can leave on the chat box, or you can raise your hand to ask the questions.
Not so much question because your talk was very nice. So very important. We have Professor Seven. So please.
Okay. Yeah, it's a very nice design. I think just want to check is this possible to open some seats for international students to join this workshop? Is that possible? Serge, you hear me?
Yes. Please, could you repeat, please? Sorry.
Okay, I just want to ask, it's a very nice design using this campus as the survey site. So I just want to know, is there any possible so we can have a talk to accept few international applications?
Yes, for sure. And important. And one thing that is to relate. So I hope that people can hear me now. So just I wanted to say that to thank really Tonka for this great talk. And I would also to say that it was his dream for many years, and now the dream starting to be really a reality, and I'm very happy. And he can really manage all of these things. This can be also an important aspect because Montpellier University is really interesting by this project, will support and will make the connection with Europe. So by having this kind of a short, for example, summer class that can allow master, PhD, and even postdoc students, and that can be also like a credit for their study, and really related to the European network. This is one thing, but also the things of this and for tropical medicine, for Kasetsart University, for all of these universities investigated, is also to make a network in Asia. And really, we're starting to discuss a little bit with Malaysia, but I think Taiwan would be also maybe another way to have this kind of site. So I want to have your ideas about this, and please, Tonkin, you can react also.
Great, thank you very much. Yeah, we'll be joined.
Thank you. Thank you. Thank you. I think Tonk is another session at the very moment because we have some conflict in terms of activities in Thailand. There is the GITMM, the Joint International Tropical Medicine Meeting. And actually also we are going to them with Professor Seven in the afternoon. So if we have no other questions, maybe we can go to the next talk, please.
Thank you, Professor Morad and Dr. Hui Shen. That's very nice for the worldwide. Like we expand research together that we can make a really nice work. And I would like to invite the next speaker, Dr. Ravadi Kamlert. She's from the Center for Disease Control and Prevention, Department of Disease Control, Ministry of Public Health, Thailand. Her professional is medical entomology, and she will present in the topic of surveillance of neglect vector-borne diseases in Thailand. She's ready right now, so I will give the stage to her. Thank you.
Hello, thank you that invite me to join this seminar. Today, I would like to share the idea about surveillance of neglected vector-borne disease in Thailand under Division of Vector-borne Disease Collaboration. The important neglected vector-borne disease under the National Program Surveillance System of Ministry of Public Health includes scab typhus, leishmaniasis, and lymphatic filariasis. An emerging disease, lymphatic filariasis, is after elimination phase situation. So we need still be follow up the remaining case and maintain positive of detection in active case finding and follow up areas. It should be less than 0.1%. And the news of emerging disease, neglected vector-borne disease are also surveillance. And all others are passive surveillance phase related to MOU between WHO and other partner countries. Leishmania is need to maintain the case under 1 per 10,000 population in this unit. Scab typhus rate per 100,000 population indicator it should be under 5-year median. Based on the passive phase of the two diseases, the surveillance system is important, covering vector, pathogen, and reservoir hosts. The area selection to plan collect our data, including high endemic area of scab typhus in Thailand. In 2020, is the northern part and present as the top 5 ranked province. Leishmaniasis is sporadic case in Thailand. So the province used to be occur of case are also our study plan as severe or visceral leishmania, so it's present in the national surveillance database. After elimination phase of lymphatic filariasis, the remaining province include 11 provinces of Thailand. We include the top 5 rank of Burma registration in each year data collection because Burma is our neighboring country that still be during elimination phase. Population group that we will focusing is related to patient occupation. The major occupation including agriculture, employee, unknown student, army, police, and others. Our plan surveillance working, we will select border agriculture village, national park area, endemic areas, and U.S. Army training station. Our plan, we will include knowledge management also. that we will covering public health staff in Office of Disease Prevention and Control, University, U.S. Army Force Research Institute of Medical Science. We would like to work and share under the network collaboration in the same major aim for people health. We will be focal point together in each institute, include tropical medicine faculty, Kasisat University, Army Force Research Institute of Medical Science, Department of Disease Control, include Division of Vector-borne Disease and Office of Disease Prevention and Control. The field covering survey part, reservoir identification, vector identification, and also pathogen detection and database. We will separate into disease as Ministry of Public Health National Surveillance System database and reservoir pathogen environment factor under the Zero Path Project website and EFRIM website also. More activity we plan to do knowledge management among health staff and villagers with different detailed information. Staff information will include biology, identification basic, sample collection, and preserve method. The process to send the sample to the laboratory. About villager information will include the transmission cycle, pathogen, vector, and host. After they know the information of disease and relation information, then let them know how to prevent themselves. The neglected vector-borne disease surveillance will strengthening needs support from every partial organizations. Try to bring neglected vector-borne disease far away from neglected fund, neglected support, neglected researcher together. Thank you.
So she tried to connect, but not sure. So I will have to thank a lot. And for us, it was quite important our collaboration between and thanks for our project for the ANR, but thanks also for this TICA project in innovative animal health, because finally we have a direct contact with the vector-borne and zoonotic disease control unit for the Ministry of Public Health. So we work directly with the Ministry of Public Health. And this was already when Dr. Rawadi starting a PhD thesis at the Mammal University. So I put back this slide that I presented so quickly. And it was a really important PhD thesis because what she did, and she was related also with the Faculty of Tropical Medicine, but with two groups from the Department of Helminthology and the Department of entomology, medical entomology, but it's close relationship with our friend from the Wellcome Trust, Oxford University, which was a really also a great support. So we can manage to have several projects together, thanks to the support of the Ministry of Public Health, thanks to the support of Wellcome Trust. So we have a project on the local level on sugar mites on rodents. And I want to emphasize this, what developed Dr. Ray for the chigger mites. The chigger mites are very important vector on reservoirs of scrub typhus. And this is completely, it's difficult to identify the species. So you need to be trained, and she was trained, but also Tonk did a PhD thesis in Liverpool, also on the subject of microbiome on related to the Orientia. So they really work together, and thanks also to Assistant Professor Sushada from Tropical Medicine. And they managed to have several innovative approach in this. So develop geomorphometric analysis using the specimens that will after be used into the molecular biology, just by adapting a technique that you can find some images on down. This is the first important things. And after to develop also innovative training to help the local Department of Health and the local vector-borne and zoonotic disease units that are present in the different regions of Thailand, to better understand the life cycle of the trigger mites, how also to identify potentially the trigger mites, or to better thing, and to relate the trigger mites with the spread of the scrub typhus disease. And this was really done intensively also in several provinces with the project, and especially in the Social Ecological Observatory of Sentong, when we try also to look not only at the rodents, when the trigger mites at the larval stage are infecting rodents, but also to try to find the ecology and to develop the ecological niche of the living trigger mites. And actually is still quite complicated and difficult to assess this task. So where the trigger mites are living, which is important in terms of the recruitment of new larvae and infection of the rodents, and of course of humans by trigger mites, and after to the spread and transmission of the pathogen Orientia tsutsugameshi. And the second thing that is starting also to work is a new project co-leading by Dr. Tonk, Kitty Ponk, with Liverpool to investigate the trigger mites that are living also in other animals, which are completely neglected. First, the birds. Birds seems to be very important. That's why when Tonk starting to discuss about the observatory, he discussed about birds. Birds can also carry trigger mites and can carry trigger mites that are concerned, that are vector and reservoir of the Orientia tsutsugameshi. But also the domestic birds like chickens, and in one of the village of Sentong, they found with Jai, the student, they found infected chicken with trigger mites that are also potentially positive with Orientia tsutsugameshi. But something that is interesting also, Michel, is that dogs are being found to be also infected by the trigger mite. So there is a really important thing. And now Dr. Rawadi, as she presented, is also interesting to better develop this kind of approach for several vector-borne disease of neglected tropical disease, which is really important. They are neglected, and we need more research on this. And this again, it shows that the Ministry of Public Health of Thailand are not neglecting the neglected tropical disease. And this is a good news. And with the help of Rawadi, she can merge and bridge the science that are developing at the Faculty of Tropical Medicine, MIDOL, the science that are developing at the Faculty of Veterinary Technology, Kasetsart University, together with the Ministry of Public Health and the district on the public health at the local level. That's why I want to thank her for a great involvement in all of this project. I speak a lot, and I saw no question. So maybe we can move to our last speaker of the morning.
Okay, thank you, Professor Moran, for the additional information about the research of Professor Dr. Revati. I think she has a great work on the with the public health a lot. And I would like to invite our last speaker in this morning, Dr. Rodolphe Hamel. He is from the Institute of Research for Development, or we know that in short for IRD, and in the subunit of infection, disease, and vectors, ecology, and genetic evolution and control, France, Montpellier, France. And he's going to present in the topic of investigating emerging vector and bird-borne viral disease. And if it's ready, so I will give him stage.
Hello, everyone. I'm Rodolphe Hamel from IRD, Montpellier, France. I would like to thank you very much to invite me to do this presentation today. And I will share with you a project on investigation on emerging vector-borne disease in Thailand. So I will first start with this slide on arboviruses, as we target this kind of virus. So arboviruses mean arthropod virus. It's a group of several family of viruses that gather the feature to be transmitted by arthropod like ticks or mosquitoes. There are more than 500 arboviruses with approximately 100 involved in human vector-borne disease. An important point is that these arboviruses are most zoonotic, meaning that a possible spread and transmission of these viruses are possible between animals and people. The impact of arboviruses in the emergent vector-borne disease is important because they represent 60% of viral emergent disease. Here in the map, you have an overview of the most known arboviruses like dengue in blue, West Nile in green, GEV, yellow fever, chikungunya virus. And as you can see everywhere on Earth, it's possible to have an arbovirus. So that represents a global threat for human health, and it's important to implement surveillance to evaluate different parameters like the dynamic and the distribution of the arboviruses, the emergence of new potential threatening viruses, and also to investigate the interaction between vector, human animals, and the virus. So it's in this context that we develop a project. The name of this project is Emergent Avian Virus Screening in Southeast Asia. This project is supported by Université de Montpellier and the LabEx CEMEB from 2020 to 2022. It's a collaborative project between France and Thailand, with IRD in France, the CIRAD, also the Service de Santé des Armées, so the French Army, and from the side of the Thai side, there are two universities, my idol university and what I like university. And this project aims to develop new techniques for identification of arboviruses using mosquitoes' excreta in order to evaluate the diversity of mosquito-borne viruses, but also the potential emerging MBVs associated with birds in Southeast Asia. We would like also to identify and try to identify factors associated with the risk of emergence at the human-animal interface. So here you have a general workflow to catch mosquito and to do the screening in mosquitoes of the virus. So on the field. We'll use different kind of traps to collect mosquitoes. We'll do the notification of these mosquitoes to sort them by pool, and we'll send and do the molecular detection and screening in laboratory on RNA. So we will extract RNA because the arboviruses are RNA genomes. We'll do RT-PCR after that, and on positive sample, we can finish the notification by sequencing of the genomes. So during all these steps, all these steps and the fact we use mosquitoes like a biological syringe to find the virus is called xenomonitoring or molecular xenomonitoring. But during all this process, some problem can appear. For example, question can appear. For example, what is the storage condition good or not to store the samples, if the condition of transportation from the field to the lab is okay or not, or how to optimize the time spent on the field, if it's possible or not to do some analyze directly of the field instead of wait for the lab to be on the lab, because of course for the molecular detection here it's better to have an access to facilities to do these things. So in this project we would like to do molecular monitoring, but not in the mosquito itself, but on its excretas, and after that the workflow will be the same. So using excretas to detect pathogens are not is not new. Some team develop this kind of analysis to detect filarial parasites and malaria, for instance, but also some colleagues already developed the screening of dengue on these excretas. Here, for example, you have one advantage of this screening on excreta is the detection, the early detection. Here you have a different graphic representing the detection by qPCR, RT-qPCR of West Nile virus, and up to 7 days post-exposition, it's possible to detect the presence of the virus. While with working on saliva, only after 15 days it's possible to see on saliva the presence of the virus in mosquitoes. The different teams, so to get the excretas, different teams develop some custom traps, as you can see on this picture, to catch mosquitoes and to get on filter paper the excreta from mosquitoes. In our project, we would like to improve all this technique with new things, new adapter or new traps to do the detections. So to do that, we develop specific traps and container to catch mosquitoes, maintain them alive with an access to a sugar, a colored solution sugar, and we place at the bottom a filter paper to get the excreta spotted on this filter paper. After extraction, we would like to analyze by RT-PCR the presence of the virus. And we have another challenge is to develop an in situ screening sequencing, but I will talk about that later. So the objective is to analyze the excreta samples in situ, not especially in the lab, to evaluate the stability of the samples in order to store well the sample prior to analyze, to get rapid results without specific facilities, and also to be able to keep mosquito alive for entomological purpose. So to do that, we developed a specific adapter with our colleague in France. So this kind of adapter here are made with a 3D printer. So the advantage is cheap, also reusable, and easy to implement. Why? Because we don't use custom traps, but we use commercial traps, and we adapt the system on the commercial traps. And like that, we are able to collect mosquitoes and excreta. To validate the detection system, we firstly work with two viruses, West Nile virus and Tembuzo virus. These two viruses are belongs to the flavivirus families, and they infect mainly birds and are transmitted by Culex genus mosquitoes. So we expose and we infect by blood feeding different pool of mosquitoes. At different time, we collect the excretas and the mosquitoes and screen the presence of the virus with a RT-PCR pan-flavivirus system. Here on the top, you have the results, the gel results of the analysis of the mosquitoes' bodies after 11, 13, and 15 days post-infection. And we were able to detect the presence of the virus. On the excretas, on the paper filter with excretas, we also were able to detect after 11 days for West Nile, both for West Nile and Tembuzo, we were able to detect the virus in the excreta. So after this first step, we would like to determine the limit of detection and if it's possible to detect the virus in excreta from individual mosquitoes. So to do that, we infect a set of Culex fasciatus mosquitoes with West Nile virus at different concentration for 4, 5, and 6 PFU per milliliter of blood. And at different time points, 5, 8, and 12 days, we collected the excreta during 24 hours. And we analyzed all these samples by RT-PCR. And as you can see on this table, we were able to detect, in function of the concentration, the virus in the excretas. And we have a dose response, or in function of the load of the virus, we can able to detect more and more the viruses from 8-10% to 100% with the highest concentration. On the right part, you have the same analysis by RT-qPCR. We measured the RNA copies per microliter of virus in function of the load of the virus and the day of collection, and each point represents one mosquitoes. So with these two set of experiments, we set up our detection system, and we were able to detect even if in one mosquitoes, but depending on the concentration of the virus, we can able to detect the presence of the virus. So an important thing was to evaluate the stability of RNA in the time, to see if it's possible to keep the excreta on filter paper a long time or not. So to do that, we evaluate the stability of the excreta at room temperature. So I collect excreta spotted on filter paper, and I keep this filter paper at room temperatures during 7 and 15 days. And after this time, I extract RNA and analyze the presence by RT-PCR. And as you can see on the gel, so here you have the pool of mosquitoes infected by 2 concentration, 10 power 4 or 10 power 5 PFU per milliliter. And at 7 days, 15 days, we were able to detect the presence of the virus in the samples. Yeah, so it's very important like that. That proves that we are able to keep at whole samples and whole filter paper at room temperature prior to analyze. So that solved the problem of storing conditions. So due to the COVID situation, we are not able to implement yet the trap in Thailand, but we performed different preliminary experiments with the trap in France to see and to evaluate the system. So to do that, we work with an institute, La Tour du Vala in France, in Camargue. It's an ecological institute located in Camargue. So Camargue is this region in France. It's the delta of the river Rhône, and it's a wide area with many swamp pools, wild fauna, and many mosquitoes there. It's also interesting to work there because the main region where the previous West Nile virus outbreak were detected in France. And here in this map, you have the Camargue area here. So there we implement 4 traps in the institute. So here on this map, you can see the different building of the institute, surrounded by field and also river swamps around. And so we implement 4 traps close to the buildings or close to the field for horses, into the forest with swamps, or here near the field with cattle. We collected the mosquitoes, and only to give you an overview of our collection, here you have after the 24 hours the collected samples of mosquitoes, and as you can see on this picture, we are able to see blue color, so blue sugar into mosquitoes, meaning that these mosquitoes have an access and get the sugar prior to drop their excretas. So here you have an overview of the filter. We collect the filter after 24 and 72 hours, and you can see the different blue spotted excretas on the filter, but also dark brown, red points like that represented the excreta from blood-fed mosquitoes that we caught. With this preliminary data, we determine, or we find some not some problem, but some issue that can happen, and particularly the impact of the rain. As you can see in this paper, the rain soak completely the filter paper, so we don't know what is the impact of this soaking on the detection. So after extraction of all these filter paper, we were not able to detect any virus in this set of experiments. So in camera. So now, what is the perspective? So we would like now to implement these traps in Thailand in 3 locations: in Nan province in north part of Thailand, in the center part around Bangkok, and in the south part of Thailand around in Nakhon Si Thammarat, with the collaboration with Mammal University, Wildlife University, and we hope to do the analysis in collaboration with the laboratory of these universities, and we will target location with birds or animals like ducks or chicken, and we would like also to implement this in situ detection directly on the field. We would like also to use new trap, because Biogen released a brand new traps. The name is BG Pro, so it's quite different than the original BG trap Sentinel. It's this kind of traps are smaller, with a power bank, not with a big battery, so the little power bank can give the power to the fan to work. So it's quite it's not heavy. Only 2 kilograms versus 10 kilograms alternatively with the battery for the BG traps. So it's light. The system is versatile because there is different kind of setting, BG trap or like a CDC trap, CDC light traps. The price is not so high, and there is also an autonomous production system of CO2 through the CS fermentation, and like that allow us to not use dry ice or to carry some bottle with CO2. So we would like to implement that also. And the one challenge now is also, beside to develop new detection of other arboviruses like alpha viruses, the challenge is to develop in situ screening sequencing protocol using MinION technology to do this detection directly on in situ on the field for rapid and identification of positive sample. But this part is a real challenge. So to conclude, I would like to thank you very much, Université de Montpellier and the LabEx CELEB for their support. Also Dr. Serge Morin, Dr. Gérard Panjaron-Paul from Wildlife University, Dr. Ronald Vargas from Mammal University, and Dr. Albin Fontaine from IRBA in Marseille, in France. Thank you very much, and thank you for your attention.
Is there any questions from the audience? And if you leave a message, or you can raise your hand.
Please be.
Or Professor Moran has to add any information.
Yes, please be free to ask questions to Dr. Rodolphe. And I would like to thank you, Rodolphe, for your very nice presentation. And indeed, what you are doing is really also really an innovative tools for investigation, for fast screening, but also for surveillance. And it will be a very, very fascinating. Can you tell us a little bit more about your next plan, please?
Yes, hello everybody. The next plan. So, as I explained during the presentation, I would like in 2022 to implement the traps on the field in the different location in order to not validate, but go further on the surveillance and on the detection, and try to evaluate the presence of different arboviruses linked with the bird fauna. So I think it will be okay to come in 2022 now, because IRD will support me, and now the border is open, so it's okay for to come. Yeah.
Thank you. There is a question from Michel for you. Michel, you can ask directly your question.
Yeah, thank you for very interesting and very exciting presentation on this new device. We can't wait to see it tested in Thailand, as you said. I had two minor questions. One is how confident are you that the device will be working as well with very different entomofauna that you may expect in Thailand? And the second one, you partly answer it, is about more specifically what arboviruses are you considering for the next phase? Which one will you be screening in priority, and asking for dengue or Japanese encephalitis or other ones, which we will be using, considering in the next steps of some of our projects?
Mm-hmm. So for the kind of entomophone that we would like to, or we expect to catch with the trap in France during the setting in Camargue, we were able to catch different species and different genus of mosquitoes, Aedes, Culex, and Anopheles. So I think we'll be able to catch the same kind of species in Thailand. So of course the trap is not specific to one species of mosquitoes, but I expect to be able to have and to be able to catch a wide range of mosquitoes. And for the second question, I don't have any. I don't expect specific to determine or to screen specific arbovirus like dengue or GEV, but I mainly hope to screen or to be able to detect arboviruses, and mainly flavivirus linked with the bird fauna, so like GEV for instance, but also some less known virus like the Tembusu virus, because I know that this virus is present in Thailand. Some researchers in Thailand report the presence of Tembusu virus in Thailand, mainly associated with the big farm of ducks. So I really like to know if in the little farm or in the countryside it's possible to find also this virus and this new virus associated with the bird fauna.
Thank you very much. I have a question to follow the question of Michel about the mosquito species. Sometimes, for example, but when we work on for example, on rodents or on big mammals, but we have maybe more samples for the excreta, we can also do some barcoding of the species. Can you think that with the excreta you can do also the barcoding of individual from the species, and to have like both a library of data on the presence of the virus, but also the detection of the species.
Yes, yes. It's another point that I don't explain during my presentation, but if you remember the filter, there is some blue spot, but also some spot, dark red brown spot, and that correspond to the digest blood finally of in the mosquitoes. So I think we will implement and set up the characterization finally of the kind of host that the mosquitoes bite to release this kind of excretors, targeting the COI. And also, I think it's possible to determine the species who release the excreta, the mosquito species. Yeah.
That is really great. So actually, you will use the mosquito as a way also to sample the biodiversity, sample also to have a better understanding of the trophic niche of the mosquito, but also in terms of transmission. So which species are connected with other species by the mosquito bite. Very important in terms of spillover on the disease ecology.
Yes, it's another kind of data that we would like to obtain this link between what we have and what we obtain in the excreta and on which animals finally or host the mosquitoes bite or both the bite previously. Yeah.
Oh, that's really great. And what is nice also, because you mentioned, Rodolphe, that not only that you are doing some field work close to Bangkok, but also in Nha and in Santong. So you will train us to train especially the young there of VetTech to go and to install your trap next week in Santong. But one thing is important that you mentioned for I think for the pilot study on the observatory of Maidol of Muka, that will be a really important stuff, because you can really I think we for sure, and Tonk and all of the group would be very happy that we can collect every year, we can monitor mosquito excreta, and try to connect that with all of the rodents, bats, and birds that are collecting at the same time and sampling at the same time. And it will be also a great activities and training activities for the student and even the staff there.
Yeah. Thank you. Yes, I'm looking forward to being able to come to effectively train and participate to this kind of year.
So we plan in June to do this training, first place of training, and we plan in normally November or December to do again a really important survey and screening. So I will give you much details about the agenda with Tom.
Thank you. Okay, thank you very much.
Do we have another questions? Not very much.
Anyone have some questions? So to the last but not least, and most importantly for today, may I invite everyone, the audience and the presenter, to turn on your camera to take a photo together. Please, thank you very much.
Yes, please. Please open your screen so we can have a virtual group photo. And I really hope next year we will be face to face. So I saw Michelle that really wait for this. Yeah, we all wait for this. Yeah, to be all together. Yeah.
Have everyone ready and technical ready? And 1, 2, 3. And hold, please hold a little bit more. And 1, 2, 3. One more time. Because we have a lot of attendees and we would like to have you all here together in the picture. So we would kindly please turn on the camera for us. I guess it's a little bit more time. 1, 2, 3. I don't know if it's in France. How do you say that for taking the pictures in France? Okay, one more time. 1, 2, 3. Okay. And I would like to invite you all for the next after the afternoon session. Yeah. The afternoon session will start at 1 p.m.
Yeah.
with Dr. Michelle Garen, which is TST.
Yes, we just start a little bit because you see that we have some problems of missing a person in this morning. So maybe better to start at 1 o'clock if you agree, Michelle, and all of your group. So we can have because I'm sure when I saw everybody will be there for this afternoon session and online also recorded, but they will be there. So if we can start at 1 o'clock. And we will have not in rush. We can be really nice and take more opportunities to have a question on exchange between us. So please be free at 1 o'clock, and we have a more longer break for the lunch.
Yes, and thank you very much for all the attendees and the presenter for turn on your camera for us. And this session, the morning session has been end, and we've finished earlier ahead of the schedule. So have take a break and have a nice lunch, and we'll meet again at 1 p.m. this afternoon. Thank you very much. See you. Have a nice lunch.
Thank you. Thank you. A lot of people miss. Terrible, huh? Wow. But this afternoon will
เปิดไมค์
ยังไงอะวนยังไงอะฝึกยังไงให้มันเล่นไปเรื่อยๆมีแค่นี้เองพี่ปื้อ